Okay, so I would like to thank the organizers for giving me the opportunity to talk. I want to thank the Kavli Foundation. I want to thank Sima and Manny, Edward and Danielle for this wonderful meeting, of course, all the speakers and the people we got to interact with. And what I would like to do in the next 3 hours is to tell you a little bit about how we can actually modify artificially with artificial molecules and artificial proteins the immune system so that it can recognize and kill cancer. So what we're going to do is that we're going to talk a little bit about the target, which is in my case cancer cells. We're going to try and define a little bit for all the scientists here what are cancer cells and how do they function. We're going to talk a little bit about a very special organ, which is the immune system. And then we're going to see how we can manipulate the immune function so that those cells will recognize cancer cells. So we are made of cells. Okay, we have around 6, 10 to the 13th cells in our body. From the moment I started my talk, each of you has generated something like 500 million cells, and no one has felt something? No. So actually, our cells are dividing and dividing and dividing. And we have many types of different cells. We have brain cells, and we have nerve cells, and muscle cells, and heart cells, and immune cells, as I will show you. And all those cells are performing their function. Those cells can form tissues. Those cells have different lifespans. Some cells will die after 4 days, like, for example, a gut cell, a colon cell. Some cells will hopefully not die, like brain cells or nerve cells. And some cells can decide to die by committing suicide if they feel like they are threatened or their existence is problematic. So those cells in our body are dividing. And basically, what is responsible for telling them to divide or not to divide? It's a very complex process, but it's mainly encoded in our DNA. And the dogma of molecular biology says that we have cells. In those cells, we have like a big Wikipedia, which is the DNA, in which we have 3 billion letters that encode different proteins. And those proteins have a certain function. And what we know is that if you start mutating, permutating, making change in the DNA, this will actually cause several processes not to function. Could be that you will kill the cell. Could be that you will make the cell do something that it was not intended to do. And I was thinking about that. I like to bake, okay, and I think that our American friends here by now have realized that there is a certain ritual taking place when we're coming back to the hotel. For the Israelis, we all line up in the lobby. And it's not because we want to complain to the hotel, but because we want to get all the parcels and all the shipments we ordered from Amazon, okay? Now, being here, I see that you understand what I'm talking about. So, being here is nice because, Amazon is really great. And I was, because of the jet lag, I'm going to show you what happened, basically. And I'm going to relate that to cancer. I like to bake, so I was thinking, okay, I'm going to order some baking, okay, accessories from Amazon, okay? So I took my computer after a long flight, and I started to type baking bread accessories, but I made a mistake. And actually, I wrote breaking bad. Now, these are the same letters, if you pay attention, but the results, you move from bread to crystal meth. And it's very problematic. And the same thing, yeah, I thought that, one of the last talks I needed to entertain a little bit the public, so the audience. So anyway, so the same process can also happen in cancer. These are very innocuous changes. We're talking sometimes, if you compare the genome of a cancer cell to the genome of a normal cell, you would find that there are only 4 to 10 permutations on what we call mutations, okay? But it can go up. And those mutations, in the case of cancer cell, can do many things, especially promote the cell division, okay, and promote the resistance of the cancer cell to what we call intrinsic mechanisms that should actually take care of eliminating that cell. And the other thing we know is that different cancer types have different numbers of mutations, or different what we call mutational load. Okay, we have cancers with very few mutations, like in the tens, and we have cancers with very, it's not working, no, yeah, it's green, but it's not working. No, you don't see that. So we're going to switch to, no, okay. Yes, we have that, yeah. So I'm going to be using both my hands. So we have also cancers like melanoma, like lung cancer, like colon or gastric cancer, which are exposed to carcinogen, which have a lot of mutations. And those mutations are very important for the immune system to recognize the cancer cell. I'm going to get back to that in a few slides. So cancer cells can form tumors. And those tumors can be pretty big and be pretty small. But basically, they can reach different organs. Okay? So you have very, very, very small tumors, like this brain tumor that was actually around 1 centimeter, a centimeter, or half an inch, which could be lethal. And you have also big tumors, like this one, the biggest tumor isolated. It's a 4 stone, so help me. A stone is what, 4-6 kilos? I don't remember, so it's 25 kilos. Okay. You make the math. We have mathematicians here. Anyway, it's something very big. Actually, this didn't kill the patient. The main problem in cancer is what we call metastasis, because metastases are responsible for 90% of the death of cancer patients. Okay, so what is that process? I just want to give you the picture how complicated it is to target cancer. Basically, the primary, what we call the primary tumor, those cells that went wrong, can reach out and can branch themselves to the circulation, okay, to the blood circulation. And just so you can get an idea of what's going on, 1 gram of tumor can release 1 million of cells into the bloodstream every day. Okay, so those cells are disconnecting from the primary tumor, and those cancer cells are traveling inside the body. It is really a miracle, if we can say like that those cells are not forming millions of what we call metastasis, okay, but only a few. What we believe that the immune system is able to recognize and stop them within the bloodstream. This is one of the mechanisms. So as I told you, the metastases are really the main problem, what we want to target. Now, what kind of treatments do we have today in order to treat cancer? So we have surgery, of course, which, if it's possible to operate, this is the best treatment. If you can isolate the primary tumor and it is possible, and the patient is operable, so this is the best modality. You have radiation therapy, which is also a kind of targeted therapy. You have chemotherapy, which is a systemic therapy, and the effects with this kind of treatment are, the side effects are sometimes very hard on the patient. We have also hormonal therapy, but I want to talk to you about a new kind of therapy that we call immunotherapy. And immunotherapy, in the broad sense of the word, would be to use or to manipulate the immune system of the patient, or to introduce in the patient new substances based on the immune system in order to cure a disease. Now, we know that the first immunotherapy, I would say, treatments were vaccines, actually, more than 200 years ago. But when we're talking about cancer immunotherapy, we actually realize that it happened only in the past 30 years. And in order to understand how immunotherapy works, we need to understand how the immune system works. So immunotherapy was really termed the breakthrough of the year in 2014 by science. And to understand how the immune system works, basically, we need to know this one thing: protect the self from the non-self. Okay, so the whole idea of the immune system is to guard the body and to defend the body, the self, against something that is perceived as non-self. And here we have a problem when we're talking about cancer immunotherapy. Because cancer, as I told you, develops from our own cells. And now the immune system, at least for several years, it was thought that the immune system do not recognize cancer cells. The immune system was supposed to ignore cancer cells because those cells are derived from our body. And we know actually that it might happen because if the immune system is allowed to attack the body, that would mean an autoimmune reaction. And we know that there are more cancer than autoimmune diseases. So therefore, it was assumed that the immune system is not able at all to recognize cancer cells. Now, the immune system, this is a family picture, which is quite exhaustive of the different white blood cells we have in our body. The immune system is formed like an army. Okay, you have different types of cells that can implement different types of functions. Okay, you have cells that are in the intelligence, you have cells that are in the heavy artillery, as I'll show you. You have cells that are commando cells, and this is another shot of what is going on in your bloodstream. You have red blood cells, you have platelets, and you have very interesting cells that we call lymphocytes. Okay, those cells are extremely important in our defense against viruses and, as I'll show you, against cancer cells. So, again, a simpler picture of what I showed you. We can say that these kind of cells, okay, white blood cells that belong to what we call the innate immune system, are cells that, like macrophages, like neutrophils, like eosinophils. When you take a CBC, a cell blood count, okay, from the lab, the results you get all those names, weird names of cells. So these are actually the cells that form our immune system. Those cells here are the cells that, when they perceive a threat, it's usually bacteria, they just bombard, they just use heavy artillery in order to kill everything around, including normal cells. And sometimes when it's not contained, that reaction is not contained, we end up with inflammation. Okay? But we have another type of cells that are very precise, and these are what we call the lymphocytes. Okay? We can call them commando units. We have three types of different lymphocytes. We have T, we have B, and we have NK cells. I want to focus on the T cells, and we have many T cells. 1% of our body is made of T cells, and different flavors of T cells, okay? So for those of you that are the same generation as me, you might remember the A-team. So you have happy T cells and sad T cells and helping T cells and depressed T cells. You have a lot of T cells. So now, seriously, 1% of our body are made of T cells. Those T cells can help other T cells to function, can kill by themselves other cells in our body that are infected with a virus or with cancer. Those cells can even restrain the function of the immune system so that the immune system won't kill normal cells and won't cause what we call autoimmune diseases. And those cells are very, very, very precise. Okay? Those cells are able to recognize a difference between, I would say, two targets, a difference that would sum up into 4 or 5 atoms. Okay? One amino acid is enough for those T cells to distinguish between a target and a normal cell. So how does it work? Okay, so you go to the supermarket and the cashier, after he or she asks, bonus card or something like that, will start scanning whatever products you're bringing to, to the cashier. The cashier doesn't know what you're buying actually. Okay, you can buy whole milk or half-half or non-fat milk. You can buy this brand or another brand. Doesn't matter. What matters is this thing. Okay, an identifier, okay, of the product, a barcode. Our cells work in the same way, meaning all of our cells, almost all of our cells. I see that there are no immunologists here. Okay. have barcodes on their surface. And it works like that. Okay, so you have the T cell. The T cell has a scanner. Okay, a scanner that we call the TCR, T cell receptor. Okay, so that TCR can recognize a molecular or protein barcode that is on the surface of our cell. Actually, each cell in our body has around 10 to 100,000 barcodes like that on the surface that can tell the immune system if it's a normal cell or a problematic cell. Okay, so that barcode actually is made of proteins that are present inside our body. Okay, inside our cells, those proteins are degraded. Okay, so and some of those proteins are loaded on the surface of the cell. Okay, so if it's a viral protein, so a viral small or a piece of that viral protein, what we call a peptide, will be presented on the surface of the cell. And if it's a cancer cell, and that was one of the main discoveries in the 90s, okay, there are, cancer cells can express, though they are mostly, they look like normal, they also express proteins that are mutated that can be I would say, cut into small pieces that would be presented, on the surface of the cancer cell or viral infected cell. The immune system can then, with the TCR, with that scanner, scan the cell and see if this is a normal or a malignant cell. Okay, just so you believe me, this is a T cell. This is a cancer cell. The T cell is just scanning.
The green cell you see here is a killer T cell of the immune system, which is attacking the cancerous red and blue cell.
Okay.
You can tell when the killer cell...
We don't have much time. So those small T cells can recognize cancer cells and can kill them. This is a cancer cell that was killed by a T cell. You can see it fires bullets, okay, protein bullets, and it will kill the cancer cell. Now we know, on the macro, we know that the immune system is in charge of defending our body. Now, if, for example, a normal cell will accumulate mutations, those normal cells will start transforming themselves in cancer cells. If, the intrinsic mechanisms in those cells, like P53 that Avi just told you about, is not working, those cells will continue to live and might be recognized by the immune system and will be killed. So that's happening, all the time. We believe, every second of our existence, we have cells that accumulate mutations that might be recognized by the immune system. But sometimes, and I can't go into details right now, sometimes for some reasons we're not really sure about them, those cancer or pre-cancer cells can evade the immune system to a point where they can develop and form tumors. I'm going to get back into that. So how can we harness T cell function? Because we know it can work. How can we teach the immune system to effectively fight cancer? So there are different ways to do that. One, I would say, is just to go and use cells that are present already in the tumor. How do we do that? That was a method, an approach that was developed by Steve Rosenberg at the National Cancer Institute, with whom I had the privilege to train during my postdoc. One of the things that you could do, especially working in melanoma patients, you can take the tumor, you can look at the tumor in the microscope, you can see that there are a lot of T cells there trying to fight, but not efficiently, and trying to destroy the tumor. You can grow only T cells out of the tumor. That's what we call tumor-specific lymphocytes or tumor-infiltrating lymphocytes, TILs. You can grow them in the lab, expand them to a huge number, like 10 to the 10, almost what we have in the body in terms of lymphocytes, and re-inject them into the body. So it works. Not in every case, not in every cancer, but especially in melanoma. And those T cells can actually go, even in the brain, okay, and locate small tumors and destroy them, and even big tumors, okay? I'm sorry for the picture, but this is a melanoma. This is a melanoma patient. This is a metastasis, okay? So this is not even the primary tumor, okay? On the basis of the neck, this patient was treated with his own T cells 12 years ago. This was last month or 2 months ago, okay? We're still in touch, and is well and alive, or alive and well, and we're very happy about that, just to demonstrate the potential of the immune system. It is his own immune system, his own cells that were used to treat him. So what do we do if we can't find those tumor-infiltrating lymphocytes inside the tumor? And now I'm coming to the artificial part of our capabilities, what we can do to force the immune system to recognize cancer cells. So, as I told you, every T cell has this kind of receptor, a TCR, that is able to recognize a small peptide that would act as a barcode. Okay? So this TCR is the scanner that will tell the T cell kill or not kill. So I was fortunate to be part of the team that did that the first time in the world in terms of treating patients with their own cells, in which we injected DNA that encoded those scanners that recognize, those receptors that recognize tumor epitopes. Meaning, we took a patient which had T cells that recognized cancer. And those T cells recognized cancer because they had a receptor that could bind to a barcode, a cancer barcode. We extracted the genetics, the DNA, genetic information encoding, okay, that receptor, and we injected that DNA into T cells from another patient that had the same kind of barcode on the surface. And we use the T cells from the patient himself with patient B, we'll call him, equipped with a receptor from patient A in order to treat his cancer. And that actually worked. Not enough, but that worked in a few patients, in like 15% of the patients. Another possibility is to use scanners or receptors that are not derived from T cells, like TCR, but from B cells. Okay? B cells are cells in our body that make antibodies. And you can use an antibody-like molecule, okay, that BCR is a cousin, a distant cousin of the TCR. And you can use those molecules, express them in the T cell, and you have two parts. One part that is actually recognizing the cancer, and the other part is telling the T cell, attack. If I bind to the antigen, to the target, you should attack. And this is the first girl that was, the first little girl that was treated 4 years ago. This is from, I'm collecting the pictures every year. She publishes, on the web, there are pictures. There are also pictures of her with Obama. That was 4 years ago, the first time she was treated. She was treated in Philadelphia by Carl June and his team. And actually, this kind of therapy work, I would say, in the case of lymphoma or certain types of leukemia, to numbers or to percentages we even dreamt of. I mean, like, it's around 90% of those kids can be cured of their leukemia. Again, it sounds like science fiction, but it's possible. And that's done only by taking their own T cells and giving them another protein, another receptor that can help the T cells home and recognize cancer cells. Again, these are nice stories, but we encounter a lot of toxicities in those treatments, and we have to be aware of those toxicities. One way to eliminate those toxicities is to target specific antigens, not common antigens, because most of the therapies I showed you so far are therapies that are applied to antigens that are expressed by a vast number of cancers. But what we know is that usually those antigens, those targets, those proteins are also expressed by normal cells. So when you teach T cells to target those antigens, you risk, okay, those T cells to attack, those T cells might attack normal cells, and this results in toxicities that can kill a patient. So one of the work we did was to focus on what we call neoantigens, only the antigens that are mutated in the cancer cells and not in the normal cell for the patient. But now you have to understand that it is a very stochastic process. And that means that every cancer patient has a different set of mutations. How can we adapt or teach the immune system of every patient to target different targets? So what we did basically was to take tumor samples from 8 different melanoma patients. we determined the gene information, the genome information, the DNA. We were able to say, okay, here you have a mutation, here you have a mutation. With the help of computers, we predicted, which mutations might be recognized by the immune system, and then we took samples from the immune system of that particular patient, and using probes that we designed, fluorescent probe, we mixed the probes that were presenting those mutations with white blood cells, with T cells from the patient. By mixing that, only the T cells that are specific to the mutated proteins would be coated with fluorescently. And then we were able to sort those cells and expand those cells in the lab. So we were creating or recreating an immune system for that patient that was built only on the T cells that recognize the tumors. And, okay, I'm going to skip that, but basically we could do that in 5 out of 8 patients. And I'm going to end up with the following. So far, I told you that one of the most important things for a T cell to recognize its target is the binding of that scanner, the TCR, to the target. Today, we know that this is not the only story, but there are other molecules that participate in the activation of the T cell. Okay, so you have the specificity, this is by the TCR, but we have also other molecules, what we call co-stimulatory molecules, that have to bind to the target cell, okay, through a receptor in order to signal to the T cell, okay, go and kill the cell, or go and, be stronger. So today we know that there are a lot of molecules that are taking place, a lot of interactions between those molecules that are taking place besides the TCR. And what we know also is that there are molecules that will activate the T cell and molecules that will actually deactivate or inhibit the T cell function. Think of that like an equalizer, okay? You have the on-off, that would be the TCR, go, no go, and then you have different frequencies to modulate the function of the T cell. Kill now, kill tomorrow, kill in a year, become a memory cell and stay with the body, kill and be killed. So we know that those co-stimulatory molecules are kind of influencing the quality of the immune response. And a lot of medicines today, like Keytruda, Adoptive, or other medicines, are blocking negative molecules on the T cell that, if the cancer cell tries to activate the brakes on the T cell, it will inhibit, or those molecules will inhibit the brakes. So minus times minus is plus, okay? So I'm going to show you a short movie so that it will make things easier. What we did was actually, because we can modify or engineer the T cell, instead of injecting a medicine, we are causing the T cell to produce a very special molecule that does not exist in nature. Think about that. You have a tumor cell that expresses a protein called PD-L1. When T cell want to attack that, let's say the TCR recognizes that tumor cell and wants to attack, the PD-L1 on the surface of the tumor cell will activate a protein on the T cell that is called PD-1, okay? And that will inhibit the function of T cell. This is very important to prevent, for example, autoimmune diseases. On the other hand, we have a molecule on the T cell called CD28 that can activate the T cell. This is a cousin of PD-1. What we did was to use the extracellular domain, okay, of PD-1, and we fused it to the intracellular domain of CD28, so that when the cancer binds to that molecule, instead of inhibiting the T cell, it would activate the T cell. Let me show you a short movie, and then we will finish. I'm going to... Okay. So last slide, more or less. Yeah, so there are different ways to engineer the immune system. I showed you only a few ways now. A lot of people, including our laboratory, is working on that. Speaking of a lot of people, these are the people we're collaborating with and people in my lab. This is a picture. You can recognize me. I'm here. And don't forget the cartoon slide. Thank you very much for your attention.